Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Hello, good afternoon, and thank you all for coming. My name is Mike Ziskowski, and I'm a program manager in the External Research Group in Microsoft. And I have the pleasure of introducing Xingyi Chua, who's an intern in our group this summer. And she is studying at the Queensland University of Technology, majoring in, or getting her PhD in bioinformatics. And she's joined us this summer to work on a project called GenoZoom. I'll let her describe the project in detail, and if there are follow-up questions after this event, or if you wish to contact Xingyi later about this effort, her contact information is actually included in the About dialog box of this application. So without further ado, I'd like to introduce Xingyi.
Thank you very much for the intro, Mike. So I'd like to first thank you for joining me at the presentation. So I'll just go on ahead. So the name of the project was GenoZoom. I do have quite a few slides, and they do cover a lot of content, but that was mainly because the slides will be put up with the application itself, but I won't necessarily go into every single point. Just a quick overview. So what was the motivation behind this project? So currently there are actually a lot of many publicly available genome browsers out there online. And we didn't want to reinvent the wheel, but we did notice that a few things of the current browsers. So they don't really scale well to the data. They don't provide seamless user experience when you're going from low to high resolutions rapidly. It's difficult to view your own sequence data in the application. So for example, the UCSC genome browser, which is probably the most popular browser out there, to view your own genome sequence, you would have to actually download their source code and then set it up yourself. And it's quite difficult for a non-Unix expert. And then they don't really support unformatted user annotations. So you'll get to see a bit of that in my demo later on. So the proposed solution for the project was to investigate how DeepZoom and Silverlight would be able to address some of these issues and make the user experience much more seamless and smooth. So why DeepZoom, or aka also known as Seadragon? So basically it was because it provided a nice way to navigate large-scale data, and it optimized bandwidth. And it did this by creating an image pyramid of your high-resolution image data, and it only downloads the tiles that you are viewing. So further advantages of actually using DeepZoom and applying it to the genome space is that it takes care of the data sampling for you because it already creates that image pyramid. With the pre-processed images, the user can pretty much select the region of interest that they're interested in, so just jump straight into the middle of the genome without downloading the entire file again. And then with the different images created as different collections, you have the potential to mix and match and create your own GenoZoom collection. So in the beginning, it all sounded very cool, very nice. But along the way, I did notice some limitations. So the major limitation of DeepZoom applying it to a genome browsing domain was that DeepZoom is primarily designed for images that sort of go with the traditional 4:3:16:9 aspect ratios. So it didn't lend itself well to the conventional genome images, which are really long and thin. So this is an example of a DNA sequence. So the demo that I'll be demonstrating is actually showing the E. coli genome. So it's a bacterium that lives in the gut. That one, for a bacterium, it's about 4.5 million base pairs, so 4.5 million characters. At 4 pixels per base pair, we're looking at a really long image. And only at 8 pixels high, it's really long and thin. So the problem with using DeepZoom in this case is that when you zoom out to have a view of the entire genome, that line is pretty much invisible, you don't see it. I tried compensating by increasing the height so that when you're zoomed out, you could still see images, but that has a performance hit when you're generating the images, and it's pretty much wasted space because everything vertically is the same. So the reason for this obstacle was primarily because of the way DeepZoom works when you're zooming into an image. It actually stretches the image horizontally and vertically, whereas the desired behavior for a Genome browser is just to move horizontally. So what I've got is an animation to demonstrate what I mean. So this is your low resolution image at the top there, level N, and then you get a higher resolution here, and then N+2. So what happens in DeepZoom is when you zoom in at N, you're actually stretching horizontally and vertically, and then it replaces the tiles from the higher resolution. And then again, and you're replacing the tiles from the next zoom level. So that's the actual behavior. The desired behavior for a browser would be at each zoom level, the height of the image is actually the same. And what happens is when you zoom in, it only stretches the image horizontally and then replaces the tiles. So I tried different ways to work around that, find out what I could do with images, how I could play with them, just to get around that zooming obstacle. So one of the trials was I have just a single hosting control, and then I dynamically lay out all the images at runtime, so specify where each of the collection of images go. So at the top there, we've got the entire E. coli genome. So it's called a gene density track. So the blue lines are showing genes in the forward direction, and the black lines are showing genes in the reverse direction. So what the red boxes are meaning is if you zoom into a region there, you get sort of more information coming up the deeper you go. So we go down to actual gene blocks with arrows pointing in the direction. You get a associated graph information, keep going down, and you eventually get to the DNA sequence. But the problem with this approach is that you sort of lose contact. You can't see all that information in one go at that resolution. So you lose what happened to the peaks of the graphs. So that was one problem. The next attempt that I did was actually go into the individual zoom layer of DeepZoom image tiles and manually tweak those image tiles. So change the sort of height at different zoom levels. So zoom levels denoted by the numbers. But what we notice in here is that the zooming action is no longer seamless. You get this sort of transparency happening and a popping type effect, and it just doesn't look really nice when you're zooming in and things are like popping out at you. And then there was came across an approach called dynamic deep zoom. So what this one was, instead of having all your image tiles pre-processed and hosted on the server, what happens is generally the MSI, which is the multi-scale image control in Silverlight that hosts the DeepZoom images, whenever you zoom and pan, it actually hits HTTP requests to the server and it comes back with image tiles. So instead of that, we intercept the HTTP requests, write our own handler that generates the images on the fly and then send those tiles back. So the classic example is the Mandelbrot plot. So that one is actually, when you zoom in, it's recalculating what you see and then popping up the patterns. And I think this is a possible solution to the problem, and it's worthwhile further investigation. The only thing I couldn't go further with it for the project was because it requires a database back end to store all the genomic data. And pretty much I was sort of weighing up doing that and time with the internship itself. So the proposed approach for our GenoZoom was we arrived at a 3-component based view. So I've got a navigation view at the top that shows the entire genome, and it's a static image, a region view, which is a zoomed in location, and then a details view which goes down to the base pair level. And I'll go into more depth in the demo. So just quickly before the demo, sort of tossing up reasons why I selected Silverlight over WPF for this project. Basically, because the selection of using DeepZoom, in Silverlight we have the multi-scale image control, which is not supported in WPF. So if I went with WPF, I would have to re-implement all the animations with zooming and panning, and then actually have to handle data sampling as well. And then with Silverlight, it's accessible by web, so that makes it really easy for biologists to share their research. And it also has the out-of-browser option, which sort of, you can download it onto your desktop and still interact with the application. But with Silverlight, there was a few disadvantages. Primarily, I couldn't use the MBF library to parse my data file to generate the image, the raw images that's required by DeepZoom. And image loading is dependent on network. So hopefully we won't see that in the demo, but sometimes the image tiles freezes, so it stays blurry. And with Silverlight, you cannot access the local file system, but there is a workaround which is to host the images on your local machine, and it still works. And without further ado, I'll look at the demo. Okay, so this is GenoZoom. So the top layer is the navigation view. Actually, I'll use the mouse, it's easier. So what I'm showing here is the E. coli genome, which is the bacterium in our gut. So the top layer is the navigation view, which shows the entire genome, blue showing genes in the forward direction, and then black stripes showing genes in the reverse direction. It's not very interesting because in bacteria they're small and pretty much the entire genome contains functional information. So if it was showing humans, you have a sparser diagram. In the region, the red box in the navigation view here corresponds to what we see in the region view. So that's from 1 to 25,000. And then the red box in the region view there corresponds to the details view. So most of the interaction and action happens in these two components here. So what I can do is pretty much just drag to a random location of interest, and this region and detail will correspond. So this is something that the current genome browsers lack with that seamless sort of interaction. When you drag to a random region, you have sort of a loading screen, not that smooth animation that's happening. So let's see what's down to the details. So I can get actual DNA bases. I don't care about the first track. I'll move that away. So in the GenoZoom, it supports unformatted user annotations. So what that means is you pretty much put it a post-it note type information. So I can change the coloring of that, and it pops up. And I can edit or delete that if I wish. What I can also do is search genes in E. coli. Please be kind. There we go. So let's have a look at that one. It's related to toxin. So again, I can add another push pin, and I can also search my annotations similar to what I did with searching a gene. Now, to simplify sort of, sorry, not to simplify, what you can also do is configure the tracks. So you can turn them on and off, and you can also add in your own custom data. So if you have some that are already processed, it will come up. But they do have to be hosted either on a local machine or somewhere on the web server. But as long as you have that HTTP URL, you can add that into there. Now, one example of how GenoZoom with that horizontal and vertical sorry, just let me restart. So while we wait, basically what I was going to mention is demo to you what happens with the horizontal and vertical zooming action. If you have graphs with peaks and troughs, when you keep zooming in, the peaks and troughs pretty much clip. So I was trying to find different ways of visualizing that sort of data, but still keep be able to sort of know what values are associated with that location. Excuse me.
What's going on with our server?
I was actually having trouble with the server this morning, so.
Where is it?
Sorry?
Where is the server? Upstairs here.
Yeah, it's actually on it in the DMZ on an external.
It's on a detail upstairs.
Okay, I'll just leave it and move on. So this was one of the issues that we noted, that with DeepZoom, you can have the image freezing up on you. One of the reasons why this was happening was because DeepZoom was set up to avoid possible DOS attacks. So if you do a lot of zooming around and panning, that sort of thing, it sort of just stops you and stops the images from downloading. So unfortunately, that is happening right now. But I'll move on. So the other thing is that you can pretty much install the application on the desktop. And if you had the net connection, you can do all the interactions that I was showing in the browser. Okay. There's also, I created a tool, the GZig, so it's a GenoZoom image generator. Basically, it's just a tool to convert the GenBanked file, put it through the MBF library to parse it and generate the raw image data, which is then put through the DeepZoom tools to create the image pyramids. Okay, some of the known issues with the application. The image generation can be slow, but if you have reference data, they don't change that often. So once you create it for the reference data, it's not too big of an issue. Data storage can be a problem because I'm creating, turning text pretty much into large image directories. But with the dynamic DeepZoom I was mentioning before, with the database backend on the server, this could potentially solve that problem. Multiple mouse events before the animation has caught up, I've seen, can have unpredicted behavior with the locations. And then with the details and range slider, so the red windows and your other views, sometimes the synchronization behavior there is a bit unexpected as well because of sort of where is the input source coming from. Few sort of disadvantages using DeepZoom in a genome browser is I can't dynamically change visuals. So what I mean by that is I can't select so for a group of genes and change the color of their genes because I'm dealing with static images. The custom data must be hosted by a web server, which I mentioned. I guess in this case, if there is a multi-scale image control in WPF, that may solve that problem. And the data size explosion from text to images is also another issue. But with images, you could potentially display much more information than you get in the text. So it's sort of weighing a trade-off there. Performance is limited by the network connections, and that again, if the images were on a local host, the performance itself could be faster. Okay, but the advantages. It does produce a smoother user experience. What I should do is actually demonstrate one of the other genome browsers that are currently out there at the moment. If you're interested, I'll be happy to demo that after. You do get quick navigation from low to high resolution. And with the tagging, it supports unformatted user annotations. So what that means is basically you don't have to conform to what the service requires. You can just put in push pins or post-it note type tags, and I have the intention of converting those tags into sort of tracks so you can save them and then upload them for future use. It's easier for the user to view their own data, so they don't need to actually download that and set up their own service, as long as they've got the data converted into the image pyramid and they can just get the URL and then put it into the application itself. And that sort of lends itself to having a potential of the user to create their own sort of GenoZoom collection. So if they're happy with all these different tracks and different information, they can put them together and then save it. And because I'm dealing with images, the application itself is sort of, in a blunt way of putting it, it's dumb to what it's hosting. It's just sort of images. So potentially you can generate any sort of image you want as long as they line up to a genome data location coordinate. And I've got a whole list of future work, but I'll just concentrate on a few. The first one is I would really like to look at creating this using the dynamic approach and using the database on the back end to see how it would solve some of the issues that I mentioned previously. Integration with Pivot, that was actually on the agenda, but we just ran out of time. And linking to external sources, so a whole list up there. And the last one is actually, I would like to compare the performance of this compared to like a pure Silverlight or a WPF version, actually drawing all those features every time you zoom and pan. So just to see what the effects are there. And to close off, just like to say a special thank you to my mentor, Mike, Simon, Bob and Vince, who I worked really closely throughout the summer and gave me feedback for the project, which I really appreciate, and a whole lot of other teams that helped make this happen. It's sort of dangerous putting a thank you slide. If you don't find your name up there, thank you. And lastly, I'd like to thank you as well for turning up for the presentation. Really appreciate that. And questions? Yes.
I saw that when you do a search, you could search on something like toxin, which
Yes.
would imply that somebody had already identified its purpose. But what if I wanted to go look for some palindromic sequence that I found in some other bacteria? Can I actually type in base pairs and have it find it for me?
Not in the prototype version. The sort of issue with that is because I'm actually, as I previously said, the GenoZoom itself is sort of dumb because it's only hosting static images. If I wanted to do that, I would also have to send the underlying base pair sequence. But if I did that, it could essentially do that. Or you could sort of do all the searches using external tools like Meme or sort of pattern recognition tools. Sorry?
And then it would tell me where to start.
Yes, and then you can upload that information as a track. Yes?
Will this tool be published outside Microsoft? I mean, like
Oh,
other
so-
people can access it?
Yeah, sorry, I should mention that. The intention is this will be open source and it will be put onto Codeplex, so the source code will be available and you can download it. The website that I was working from is actually MBF server. It might be a temporary link. We haven't worked out if it's going to be hosted somewhere yet. That's going to be, I'm going to talk to my mentor after that.
We'll end up keeping it there at a minimum, so people outside Microsoft can access it, demo it, as well as the source code on GitHub. There'll be a document as well as this presentation.
Yes. So there'll be a technical document which goes more into sort of the classes and structures and all that stuff as well.
How to build.
Yeah.
Could you comment a little more on the problems you had with image generation? Because I have some of the same problems. The DeepZoom image generation, how long it takes? Because obviously I think a lot of us who deal with DeepZoom have those problems. What kind of possibilities are there that might help that?
Okay, so with image generation, my sort of bottleneck was actually producing the raw images that would feed in as input into DeepZoom itself. So it wasn't actually DeepZoom making the pyramids, that was all right, but it was actually generating 4.5 million characters and then all that. So I was looking at 1,500 images. And then for each track, I was looking at 1,500 images, and then those were the bottlenecks. So different ways of trying to do that was parallelizing the code. I think it depend on your underlying structure. At times, it saved me time, improved in performance. But at other times, because it was a contiguous block, I needed to know what images were in my previous block, so I couldn't do that in parallel. But I think that just needs a rewrite of the code itself.
So have you had a chance to have anybody in the domain really look at this and see, is it significantly better? Some of the ideas and the transitions, the animations help people maintain context when they're browsing, or what other feedback have you had a chance to get from either anybody back in Australia or wherever?
So short answer, no. Longer answer, earlier sort of mid-internship, I was doing the user experience studies. At that time, I had a very, very early prototype version of the GenoZoom, and I had sort of handed out to people to see their reaction. One person sort of liked the whole animation, smooth navigation, so that was positive feedback. But I think generally, we didn't see that many people, and generally it just really depends on what they were working on, because the trend is that the biologists tend to stay within a specific zoom level. They don't really go from all the way from a genome down to the DNA. They tend to sort of go either the top to sort of the navigation and the region view, or the region view and the details view. So, yeah, I think need more sort of user feedback on that. And then the other, I suppose, I did send a link to David Heckerman, so I'm hoping to get some feedback from him as well.
Do you think this will scale to do the human?
Yes, because basically you're just doing images. So I'm thinking theoretically it should just scale. The only issue with human is you're going to have to spend probably a day to produce those images. Yes, and storage, actually, that's going to be an issue. So an example of storage was the E. coli GenBank file is only 13 megs. Those image pyramids came out to be 12 gigs. So, yeah. So,
hard drives are cheap.
yes. So that's sort of why I'm sort of really looking forward to doing that dynamic deep zoom approach to see what would happen there. But again, as I mentioned, in images, you can potentially put in a lot more information in there than what is in a normal sort of GenBank file. So I could, for example, color code my genes based on function or cog categories or sort of different domains, that sort of things. And that's still a single image, but I've got a lot more information in that single image than the normal text-based source. No other questions? Okay. Oh.
So aside from research purposes, can you
Yes.
see this being used for anything else?
So one of the thoughts throughout the internship was actually this could lend itself to an education domain. So something like the GenoZoom. I'm not sure if you're familiar with the GenoZoom, but basically, yeah, it might lend itself well to the education domain. So in that scenario, one could do maybe a cooked up tutorial that goes out from, if you want to aim it at high school level or at sort of undergrad level. So it cooks up from, this is your entire organism. Let's concentrate on maybe a dystrophin gene, so it causes some certain diseases, and then it goes down to maybe the protein level. These are the domains we're interested in. Mutations in the DNA causes this sort of diseases, that sort of thing. So it goes down straight down to the DNA level. So that type of tutorial may suit for this type of application. Okay, no other questions then. Thank you very much.